Hi, everyone. It's great to be here with all of these friends and family of friends. It's nice to see you all, and I hope to chat with some of y'all soon. So Manuela has been saying that I should kind of give a TED Talk on some of my recent experiences in grad school, so I guess this is the closest I'll probably get to that. So I'm honored to be here. So I work in a lab that is really concerned with studying dendrite regeneration and understanding how dendrites regenerate. Now, if you're not familiar with the term dendrites, brief little neuronal anatomy lesson here. I like to use my arm as an example a lot of the time. So if you think of the palm of my hand as like the cell body, that's kind of where the nucleus is, where the DNA is of a neuron. You can think of my arm as the axon, so that's kind of like the signal sender of a neuron. And then you can think of my fingers as the dendrites, or the signal receivers of neurons. So neurons are connected in these networks, and they kind of transmit information along these processes. So we're really concerned in the lab with how the dendrites regenerate. My project in particular is really interested in how neurons sense that their dendrites have been injured, and also how they mount a differential regenerative response to dendrite injuries. So the way we do that is we actually work with fruit fly sensory neurons in the lab. So if there's one thing you take away from my talk today, it's that I'm the fruit fly guy. I feel like that's kind of what I'm known for in this community. One of the only ones that does neuroscience and fruit flies. So I'm really interested in how neurons use actually calcium to detect that they've been injured, the same calcium you get from milk. And what I've been able to show throughout my graduate work is that different neuronal cell types are actually better or worse at sensing that they've been injured using calcium. And they also actually mount different regenerative responses. So they have different ways of like defending against some sort of injury. Now, this is really important because what we see in terms of disease is that dendrite shape actually changes before, in some cases, it changes before the onset of symptoms in certain neurodegenerative diseases like Alzheimer's, Huntington's disease, Parkinson's. So it's really important that we get this down, that we understand how neurons are sensing injury, that we understand how neurons are mounting a regenerative response to these neurodegenerative diseases. More recently, I did a really unique and really fun experience where I was able to go out to Boston and do a big internship with a huge pharmaceutical company, Eli Lilly. I don't know if you know them. They're like the big insulin makers. They also made like their own form of Ozempic recently. And that was a really great experience. I think the best thing I took away from it is that it really reinvigorated my passion for research, because all these people that are making medicines and making big bucks to make medicines, they actually really look at our research and really consider it so important for the stuff that they're doing in order to translate our findings into medicine. And that really reinvigorated, like I said, my passion for basic research and my passion for what I'm doing in my PhD lab. And all the skills that I was able to learn and kind of like that realization that I had while I went away for an internship, I really was able to bring that back. And now I'm really pushing what our lab is comfortable with and actually trying to move away from the flies. I love the flies, but they can't answer all of the questions that we have regarding dendrite regeneration. So something really interesting is flies don't have dendritic spines, and dendritic spines are really important for learning and memory uniquely, at least in terms of the dendritic compartment. And so one direction that I really want to take the lab in is to look at dendrite regeneration in cultured hippocampal neurons. Neurons that are derived from basically the seat of learning and memory in our brain, and really ask questions like, can mammalian neurons regenerate dendrites? That hasn't been shown in our field. If they can regenerate dendrites, can they regenerate dendritic spines, which suggests that if a neuron has been injured and it regenerates, we can maybe restore some of those memories that were lost. And then can they reform functional connections? Because we also don't know that in our system, because we're working in the periphery and there's not really any neurons upstream of that. So with that, I'd like to thank my amazing mentor, Dr. Katherine Thompson-Peer, who's here today, for all of her support throughout my graduate work. The YASAs for this really special environment that they've been handed, but also have cultivated and maintained and really grown so much throughout the years. Thank you to you guys. And then thank you to the family and friends and colleagues of Jared Roberts for making this award possible. Thank you.